So the next section talks about modern classification systems. So classification systems help humans organize all of the other organisms in the world. However, the method of classification has had to change and adapt over time and grow itself. And many species, as we've moved along, have had to be reclassified. Like, for instance, this lovely butterfly here. It says that before, early taxonomists would have put them in different species, but in fact, it's sort of like humans where they maybe have slightly different, fuller wings or like a more streamlined shape, but in different coloration. But this is actually the same species. So just like you can have a variety of humans or dogs or cats, you can have a variety of this butterfly. And so some things have had to be reclassified based on, as more information has become known. So there's three basic classification concepts, and I'm going to talk about each of these. So I'm sorry this is a boring picture to look at, but it's a helpful chart to have. So the typological species concept is basically based on what you look like. So based on your actual traits that you had, they would take one or a few example organisms and say, is it about like these? And so then when they would find something new, they would always compare it to those examples that they had. Well, obviously the limitation on that and why it's not, the limitations is like why this doesn't work well. And one of the reasons that this doesn't work well is that um, obviously there's variety. You can have a number of different things about a horse or a pig or a chicken that look different, but that doesn't mean they're not the same species. And so the benefit though is that there's been a lot of people for a long time keeping good records. So there's a lot of good data on this. So it's still pretty helpful. The next is the biological species concept. This is pretty similar or pretty simple in that can two things reproduce, like breed and have a fertile offspring? And if they can reproduce and make a fertile baby, then they are the same species. The problem is this obviously doesn't help with extinct species. Like, what were these dinosaurs breeding with? We don't know for sure. You can't know that. And even like more recently, things that go extinct really quickly that, we just now discover it and it's already extinct, we don't really know. Um, and then there's some things like a dog and a wolf that are clearly different species, yet they can breed and produce a fertile offspring. So this is correct a lot of the time, but it's not helpful for extinct stuff, but it's still used a lot. The next one is the phylogenic species concept. So this is looked at the evolutionary history, like the adaptations and things over time, how things are related. And the problem with this is it takes a long time to figure that out and to work with that, to look at the DNA before all that information is known. So this accounts for extinct species and it considers molecular data, so it's very helpful. It's just, it takes a long time. Okay, so here's a really cool dinosaur bird and another bird, okay, that you would maybe see today. So these two animals from fossil evidence, we can see that they both had hollow bones. They were both generally shaped like a bird, so they probably have some things in common. So these are called characters. And characters are features that um, are morphology, like body shapes, like either their anatomy or their embryology or their DNA that can be similar or different. So there are some methods used to determine phylogeny or evolutionary history. So phylogeny is like the evolutionary history of a species. And one of those methods to compare is used to construct patterns of descent by using characters. So patterns of descent, like how things were different and changed over time. So, morphological similarities is how two things have a similar body plan. And here's an example. In a plant, I have a picture that you can see. Here's some palms. These are probably date palms, but you can see how they're actually different from each other. And their leaves, and while they look somewhat similar, their leaves also are somewhat similar, but they are a little bit different. And their seed pods that grow off in different ways, and their seeds go and grow the seedling differently too. But you can see a lot of similarities here. So probably the same species, or probably a related group of species. Though they're different species, they're probably related. You can also, besides morphology, you can look at DNA to see how related things are, and we talked about that in the last section. But if you look here, you can see like the chimpanzee, the gorilla, and the orangutan all have this blue in their DNA banding pattern, but the gorilla had a red, and the chimpanzee and the orangutan have the purple that follow. But then here, the chimpanzee is slightly different in that it has a red right here that nobody else has. But in some big ways, like the orange, or this blue and orange, or this red, blue, and green, they are the same. So looking at how similar or dissimilar DNA is really helps us to know how closely related two things are. So if you had say 65% or so of your DNA similar to something else, well, then you're not that closely related to that organism. But say you had 95% similarity, then you're going to be, fairly related to that organism. And that's basically relatedness, and how humans are sending off their DNA to have DNA tests now, and they can find different relatives that have also done the same thing, sometimes people you don't even know, and you can see how similar their DNA is. It's very interesting. So, here's like the different types of elephants. There's the African that lives on the savanna, there's the African elephant that lives in the forest, and then there's the Asian elephant. I am going to encourage you to also look up the Bornean pygmy elephant, which is not listed here. That one I worked with in Borneo, and they're actually working on DNA work with that to see if they can get it classified as a separate species, because it's much smaller. It's a pygmy elephant. The babies are like knee high, and the adults are like my height. They're adorable, and if you like elephants, go look that up. So this is the molecular clock that I was talking about, and this is a picture of a molecular clock. So a molecular clock, um, and I'm not in love with this picture, but it shows that there's an ancestral gene, and here it went through two mutations on this one and two mutations on this one. Okay, and I think mutations happen usually a little bit slower, not necessarily two at a time, but um, that's what this picture shows. And then this one further got another new mutation, and this one went through a few more mutations, and then slowly your DNA changes over time. What that picture is trying to show is how these maybe had a more common ancestor at some point, but their DNA got increasingly dissimilar to where they were different species. I want to point out that the misnomer, or the incorrect term of using this animal evolved from this, that is an incorrect thing to say. One did not evolve from the other one, it just got more dissimilar over time. Okay, so the next thing that we're going to talk about is cladograms. So here in the cladograms, this is a cladogram, and you see these sometimes. And I want to first look at the lilies. In the lilies, the characteristic that it has is it's a flower. Now, only the things above this line will be flowers. Well, the only thing above the line is lilies, so that's the only one that's a flower. Conifers, which people use as like their Christmas tree, and ferns and mosses, none of these have flowers. So if you look here at this descriptor of seeds, the conifers and the lily have seeds, but ferns and mosses kind of have like spores, so they don't have seeds. So everything is up here, this thing is the most complex, and whatever is down here is the least complicated. Why are lilies the most complicated? Because every one of these descriptors along the way all match for lilies, but like the mosses, they only have one thing on the cladogram describing them, which is the anchors or roots. So they only have one thing describing them. And then I also want to point out that on a cladogram, if you go all the way down, you can find the thing that links all of them. And so they all had an ancestor with chloroplasts. So like something that could make its energy from the sun. So the purpose of a cladogram is that it helps us analyze these types of relationships between each other. So it's all based, let me ask you a question. If we have mosses, for example, how are mosses right here, how are they different from ferns, conifers, and lilies? If ferns, what do ferns, conifers, and lilies all have that mosses don't? Well, the answer would be go up one rung and see what it is. It's vascular tissue. So mosses don't have vascular tissue, but all of these have vascular tissues. But you couldn't say seeds because the ferns don't have seeds. So if you're saying what makes mosses different from all of these, it's that it's the vascular tissue. So you go what's the next thing up. All right, the last thing is that here's the domains and how they break down. The domain eubacteria is here, and then the domain archaea is here, and then the domain eukarya is here, and that breaks into 4 different kingdoms, and we will talk about that in the next section.